All right. Let's call this coupling exergonic reactions with endergonic reactions. And let's sort of give it a subtitle, ATP. So we were finishing off the last video, we were discussing the concept of the exergonic and endergonic reactions and using Gibbs free energy to determine which was which and which would be spontaneous and which wouldn't. And spontaneous reactions, because they don't require a constant input of energy, will just proceed once they've been kick-started for on and on. Whereas the non-spontaneous ones require energy to be added in steadily all the time to keep them running, which means they sort of work counter to the laws of thermodynamics, which say that everything proceeds along a spontaneous path towards more entropy, more disorder, right? But the reason these endergonic reactions can happen is because, at least in a biological setting, we can utilize the energy that is released by the exergonic reactions and use it to power or to keep running an endergonic reaction. And to do that, we do something called coupling. We couple reactions together. And of course, one of the molecules that couples reactions together is the ATP molecule. It's the link. In other words, it allows us to sort of borrow the energy or capture the energy from an exergonic reaction and use it to power an endergonic. There are others, and we will see some of those other energy-carrying molecules later. But for now, we're just going to deal with ATP because it's probably one of the most common, and it's going to play very nicely into our next section, which is going to be studying the oxidation of glucose. Okay, so first of all, let's get to know ATP and what it looks like. So let's draw ourselves a little picture. Let me just go here, get some space, and we'll draw a picture. So here's what ATP looks like. So let me get a thinner line. So it's going to be a fairly large molecule across the page. And essentially, it's also going to go up a little bit. So start down so you have room to go up on the right side of your page. I'll start down here. Whoops. And of course, this is our phosphates. Adenosine triphosphate is essentially what we're talking about. So we need to have 3 phosphates. That little tick I made is deceiving. That's supposed to be a charge. So one more time. I'll try not to make too many corrections here. There's 1 phosphate in the bottom. And these little charges, maybe I'll put the little charges on in a different color afterwards. Here's a 2nd phosphate. And here's a 3rd phosphate. Okay. There's the 3 phosphate molecules. I'll put the charges on later so I don't have to keep changing my line size. This last O, though, is connected to a CH2, which is part of the bond here. And then what we have is sugar. There's O at the top. It's a ribose sugar. So it has 2 OHs down below. And it has another bond here. There's just an H on. I'll put these H's in today just because they remind us. And then at the top, we have to do something here. We have to squeeze in, and unfortunately, it's going to be a little bit weird, but I'll do it as best I can. I'll have to go small. Carbon, double bond, nitrogen, carbon, carbon, double bond. So it's a little pentagon shape right there. There's a little H on this guy to fill him out. And then there's the hexagon shape. double bond there. And then down below here, there's a C with a little H attached. A double bond down below here to an N, and then this to here. You might recognize this guy. You might recognize this section here as the molecule adenine, one of the nitrogenous bases. And then you should recognize the molecule down here as a ribose sugar. And then these are the phosphate groups. Sure. We're just going to add one little thing we forgot. Right here, this little C is missing an NH2 to fill out his bonds. So that's what adenine is supposed to look like. Add a what? Yeah, oh yeah, I promised to do that too. I'll do that in sort of red. The O's have the little charges, or they have little H's attached, charges like this. They tend to remain ionized in solution in water. So those would be the little charges on the O's. And so we have here sort of a three-piece molecule, which is basically called a nucleotide, right? It's just like a nucleotide that makes up DNA, except this particular one floats freely in the cell. And these three phosphates, rather than join up, remember how the phosphate is sort of the link between the DNA structure. Instead of joining to another nucleotide, it just joins up to these three phosphates. Okay, this side of the molecule, if let's see, how can I do that? If I take the adenine molecule and the ribose molecule together, those make a nucleoside called adenosine. And so now you can see where the name comes from. Adenosine, which is the two molecules on the right, attached to three phosphates, adenosine triphosphate. So that's our ATP, adenosine triphosphate. Or for short, ATP. Now, the sort of action site of this molecule is these three phosphate groups here. Each phosphate that's added to the molecule adds energy to the molecule. Remember, if you are increasing the complexity of something, making it more orderly, then you are decreasing entropy and increasing energy. So with three phosphates attached, the ATP has more energy than it would if it had only two or one. The nice thing is, though, is that those phosphates are relatively easy to dislodge. So in this state, the molecule contains energy in the bonds between the phosphates. What ATP can do is it can, it can lose a phosphate group. Hydrolysis. In the process of hydrolysis, it can be chopped off. And then this phosphate that floats free can then be reattached to another molecule. We call that phosphorylation when a phosphate is attached. And we're going to see that. That's the way that the molecule transfers its energy. The energy that's stored here in this bond is, if this is released, it's attached to another molecule, which then creates a higher energy state in that molecule. And so what we commonly see is a little reaction where ATP breaks apart by hydrolysis into adenosine diphosphate. The D stands for di, which would be two phosphates left, plus a phosphate. We often refer to it as inorganic phosphate, and we draw it with a little subscript I. That's now free-floating. But this free-floating inorganic phosphate doesn't stay free-floating for very long. One of the things that happens in biology when we start describing reactions is we have all these intermediates, we call them. So this breaks up into these two intermediates, but almost immediately, these intermediates will react with other things. That phosphate might attach to another molecule, giving it a higher state of energy, or it might reattach to another ATP in another reaction, coupled with the exergonic oxidation of glucose, as we'll see. So remember, everything's happening in a soup, right? So sometimes we have these intermediate molecules and they don't last very long. And the phosphate wouldn't last very long by itself because it would ionize in solution, and those two charges on those oxygen molecules would make it quite reactive and want to stick with something else. So we often see this. And of course, what I should do to be entirely accurate is I should change this arrow to a double arrow back and forth. Maybe I'll put a red arrow on the top this way, but I'll put a blue arrow on the bottom this way. And though we could recognize that the reaction, the red reaction going to the right, is a spontaneous exergonic reaction that will release energy. The energy is released in that phosphate, which will then be reattached to something else and transfer the energy. Whereas the blue is an endergonic reaction that is not spontaneous and requires an input of energy. We're going to see very shortly how glucose can be used to cause this blue reaction to run backwards. The exergonic oxidation of glucose will be coupled to this reaction so that we can generate or make adenosine triphosphate out of these two things on the right. All right, so that's our ATP molecule. It turns out that we can actually measure the amount of energy that is given off here. The red reaction that goes this way is exergonic, so it releases energy, and it actually releases, in laboratory conditions, we can get about 30.5 kilojoules per mole of energy. In laboratory conditions. That's with standard temperature and pressure and all of those things. But inside a cell, things don't always follow standard temperature and pressure. So they're a little bit different. And inside a cell, because of the conditions, we can actually get closer to about 57 kilojoules per mole. So the amount of energy that's available inside the cell is actually more than we can achieve by doing this in controlled conditions in a laboratory, which is kind of interesting. Okay. So in a cell, when this ATP hydrolyzes and releases its phosphate, there is going to be somewhere between 30.5 and 57 kilojoules of energy that can be released, depending on the conditions at that immediate moment within the cell. All right. Now, what's interesting is this. If that P just kind of pops off, right, two things can happen. Either the P basically just is removed, and the energy that we get is basically just heat. Or the P can be attached to something else. Oops, let's spell properly today. Not that it really matters, but it matters. Attached to another molecule. In other words, we say it's coupled, and that can cause other things to happen, coupled to a reaction. So your body can utilize both of those. When it's cold outside and you need to warm up, ATP energy can be converted directly to heat simply by letting this reaction run in this direction. However, most of the time, we want this energy to be attached or coupled to something else, so this phosphate is going to go somewhere and it's going to do something in another reaction. Okay? So the three things that ATP can be used to do are mechanical work, where the ATP phosphate is attached to something and it changes its shape and literally causes it to move or bend in a certain way. Okay? Mechanical work is the changing of shape or movement so the phosphate can attach and cause things to do that. One of the examples is that in the cell, there's a network of cytoskeleton, right? These fibrous things that run through the cell, and organelles are actually attached to them by protein molecules, like little feet, so to speak. And when you add the phosphate to them, it causes them to shift or change in their shape and move. And so this little thing can actually wiggle its way along this by adding the phosphate and removing the phosphate and adding the phosphate and removing the phosphate. You get this little robotic movement or jiggling of the proteins, which causes it to essentially walk along this cytoskeletal network. And so ATP can be used to move things around in the cell. The phosphate adding and subtracting. Like the which? Myosin and actin. Yes, it probably is very similar to that. I don't remember precisely what the entire biochemical pathway is, but I think the ATP is involved there in changing the opening and closing protein gates on the muscle cell membranes, which allows the flow of calcium ions back and forth across, which then changes causes the actin and myosin to contract. So we could look up that one if we want to know the precise role that the ATP plays in that pathway. But that is another example. It's a literal movement of something. The other thing is it can do transport work, as I just mentioned. And that's, well, it's kind of the same thing. It basically allows molecules to be transported through protein gates on cells. So across cell membranes, essentially. It's still technically mechanical work. All work is mechanical at some point. It's all force times the distance. But in this case, the phosphate would be attaching and releasing from a protein molecule on a membrane. So if you had a membrane and then you had a protein molecule inside of it, here's my phospholipid bilayer, roughly drawn. And you have a protein molecule here inside, then the ATP, as it attaches, you get ATP, which becomes ADP, and the phosphate gets attached like this to the protein, changes the shape of the protein, and opens or closes the gate. And then when the phosphate is then detached, it goes back again. So you can regulate by having ATP deliver its phosphate here as transport work. And then the other thing it can also do is what we call chemical work. Chemical work is done when we transfer energy from one molecule to another. It's basically phosphorylation. Phosphorylation of a molecule to raise its energy. Raise its energy so that it can then carry on and do what it needs to do. We're going to see that in our next video when we start talking about glycolysis. Okay, so that's the wonderful ATP molecule. And as I said, there's a few others too. We're going to learn about NAD and some other ones when we get into glycolysis, but we'll address them as they come. This is just a good example because ATP is perhaps one of the most common. And again, this number I gave you, we're not going to be doing too many calculations, but it just gives you some idea as to how much energy is released from there.